And Dr. Pitkow, thanks for joining us. We're just having our panelists introduce themselves. So if you want to go ahead and just brief introduction, that'd be great.
Sure, yeah. Hi, sorry I'm late. I'm Zach Pitkow. I'm a faculty at Baylor College of Medicine, and I have a joint appointment at Rice in the Department of Electrical and Computer Engineering. I'm a computational neuroscientist, so I think about how the brain works. I think about thinking, and I use math. I'm a theorist, so I use methods from math, statistics. My background is in physics. My PhD is in biophysics, and we like to try to merge the cognitive perspective with the neural one and try to understand how the brain actually implements interesting computations through the neural machinery that it has. Is that a good short introduction?
That's perfect. Thank you so much. So basically, we will have each of our panelists present what they have for us, and we'll have a short little Q&A where you guys can ask them questions about anything. And yeah, so Dr. Leal, if you want to start with your presentation, we can get started.
Sure. Okay, can you see the slides okay?
Yeah.
Hear me okay? All right. All right, so as I mentioned, I started my lab a little over a year ago. We're called the Neuroscience of Memory and Aging Lab. So those are the two main areas of research focus in the lab. And we're located in the BRC, in the Bioscience Research Collaborative, right off of campus. Although right now everything, of course, is remote, but the laboratory space is there where we typically conduct our cognitive and behavioral studies. We perform our MRI studies at Baylor, at the Core for Magnetic Resonance Imaging, or CAMRI. So they have two scanners that we use for research purposes. And then we also do PET imaging studies, where we look at different proteins in the brain. And this is specifically targeting the aging population and looking at some pathology that's associated with Alzheimer's disease in cognitively normal older adults, and try to understand how that might be impacting memory function as well. And that's done at Houston Methodist. And so the lab is really focused on understanding human memory. And more specifically, as I mentioned in my introduction, we look at factors that are modulating human memory, such as emotion, stress, aging, and depression are some of the main focuses of the lab. But it can be pretty widespread in terms of anything that can impact memory. We're sort of interested in seeing how that impacts behavior and how that impacts the brain. And so we use different techniques and methods to look at these different types of cognitive functions. One, as I mentioned, we look at behavioral data and using cognitive testing in the laboratory. So typically this is done with participants. Typically a lot of undergraduates who are in psychology courses will conduct or be our participants in these different SONA experiments that you guys have requirements for in your courses. And so you might have participated in some of our studies where you are in front of a computer screen and you're looking at different types of stimuli and you're asked to make judgments about them. And in our case, we typically ask you to remember different things. We also do neuropsychological testing in the lab, which is a different set of paper and pencil tasks that allow us to look at a more broad cognitive function like attention, processing speed, and things like that, in addition to memory. We also use neuroimaging methods, as I mentioned. So we use MRI and PET to look at brain activity while people perform different types of memory tasks in the scanner, as well as look at those proteins that I mentioned that are associated with Alzheimer's disease using PET imaging. And then we typically apply both of these types of methods to clinical populations, so people that are experiencing depressive symptoms or older adults who might have some memory impairment going on. And if anyone has any questions during this, feel free to interrupt me. So just kind of a general overview of what the lab does. So we focus on developing new cognitive tasks in the lab. So undergraduates could potentially get involved in that side of the research. We also look at these advanced imaging methods to understand brain function. And we have ultra high resolution MRI, so we can look at really tiny subfields. It's hard to see in here, but we try to look at the highest resolution that we can possibly get in the brain to be able to test questions about different memory regions in the brain and how those underlie the performance that we see on these different cognitive tasks. And then we apply both of those to translationally to different types of populations with the hopes of understanding what is going wrong in the brain in different populations that might have memory impairments, such as aging and Alzheimer's disease. And if we can modify that in some way with treatments or some sort of interventions. So just a brief background on what we kind of focus on in the lab and what we study. We focus on episodic memory, which is the what, where, and when of memory. So what happened, where did it happen, and when did it happen? But we also are interested in why we remember certain things. So remembering significant experiences in our lives versus things that are kind of more neutral and that might not be as important to remember. And so we really, a big part of the lab is focusing on this why component of memory. And I like this quote from William James, who said that some events are so emotional as to leave a scar upon the cerebral tissues. So we often think about these emotionally or significant experiences as something that's really important to remember, and it's almost like it's, you know, a scar in your brain, and it's really kind of locked in to our memories if it's something that is important to remember.
Can I comment on that real quick? Because it just reminds me of something that Terry Sinowski told me once. He's a scientist at the Salk Institute. And he said that all memories are scars, which I thought was a really, like, what do you mean? The actual matrix that composes the outside of the neurons, like all the axons and all the shapes, are made of collagen, which is the same material as scars. So as you learn and you make all these little patterns of what connects where, if that neuron dies for some reason, it leaves a tube behind, which is the scar of where that memory used to be. So I thought that was very beautiful, and I'm, you know, it's neat to connect it to the other thing that you just said, where memories are scars, especially for challenging events or dramatic events.
Yeah, that is really interesting. And also in the case of Alzheimer's disease, there are, when the cells die, there's evidence of, you know, what's left from that, which are the plaques that are evident. So it's, yeah, interesting that it's kind of a nice analogy of that. Okay, so within the brain, so this is kind of just a general idea of what we study with memory, but within the brain, we focus on the medial temporal lobe, which is the main set of regions that are involved in episodic memory. And so we studied the hippocampus, which is this long green structure here, the amygdala, which is involved in emotional memory. And then there's some other regions as part of the medial temporal lobe as well that are important for memory processing. And so with imaging methods, we're able to look at these, especially the high resolution imaging methods. We can look at these different structures and try to understand memory, the neural mechanisms underlying these different types of memory functions. So just to show you an example of one of the tasks that we've designed, this is an emotional memory task that we've made where people are shown these different types of scene stimuli in front of a computer screen, and then we test their memory for those images at a delay, either immediately or 24 hours later, sometimes even a week later. And then we try and see how much information you can retain if things are identical to what you've seen before, or if something is similar but not exactly the same to something you saw before. And so by doing this, and I won't get into the details, but the different parts of the hippocampus underlie different parts of this type of memory processing, and how much detail can you remember from these really similar ones versus these items that are just the same thing that you've seen before. So we develop all sorts of tasks like this that we give to participants in the laboratory, and as an undergraduate research assistant, you would be most likely administering these types of tasks to the participants. I also mentioned neuropsychological testing that we do to kind of give us a more general sense of cognitive functions that aren't as specific as that task that I just showed you. So we can kind of get a gauge of cognition more generally. This is an example of one where you read a list of words and then you ask people to tell me as many words from that list as you can remember. And so it's a different way of testing memory functions with these kind of standardized batteries of tests that people use all around the world so that it's comparable across different studies. We also use questionnaires in a lot of our studies to try and understand things like depressive symptoms and how that impacts memory, other sorts of questionnaires that link to these, that might impact memory function. So this is an example of a sleep questionnaire that we have to try and understand how sleep impacts memory, since that's so important in consolidating our memories that we have throughout our day. So we try and get as many of these types of measures as we can to try and explain why people's memories vary so much and if these things could potentially explain it. So in terms of the imaging methods, this is a zoomed in version of the brains that I showed you earlier. And so we're able to really look at these tiny regions in the medial temporal lobe to be able to look at activity in those regions while people are performing the tasks that I showed you before. And so we can look at these little regions of the hippocampus and quantify how much activity is happening in these different regions, depending on if you're correctly remembering something or if you're if you're not remembering it accurately. And we can look at these different subregions using these methods. So as an undergraduate research assistant, you could be involved in looking at some of this data. It takes a long time to analyze, so it's typically not something that, like a project that can be done all independently. But if you were interested in getting MRI experience, it's great to be able to go to these scans and kind of look at brains as they are getting acquired on the MRI scanner. And some, you could potentially be involved in some of the analysis side of things as well. And then lastly, the translational applications of the lab. Mostly we focused on depression in the past, where there's a negativity bias in memory, often associated with depression. And I'm not gonna go through the specific results here from one of our studies, but basically individuals with depressive symptoms seem to be impaired on some of those memory tasks that I've talked about before. And they also show an enhancement in negative memory for a specific kind of memory. And so it's really powerful to be able to develop these types of sensitive tasks and looking at things like symptom severity in this case. So the more depressed people are, the better they remember negative details in this case. We also apply the tasks and all of these different methods to the aging population. So we've found that on those tasks that I mentioned, older adults, as you get older, they start to get worse and worse at being able to do these types of memory tasks that rely on really detailed memories. And a lot of this work is based on animal models, actually, of what's happening in the hippocampus and the medial temporal lobe in aging, and can we look at that in humans with this higher resolution MRI. We also look at if older adults are on this normal aging trajectory versus maybe they're actually on this path to developing Alzheimer's disease or dementia. And so we use PET imaging to try and understand this. So we look at people that are pretty much in this range here in this preclinical phase, where they might not have a lot of memory impairment, but they might have a lot of amyloid or tau in their brains. And so we try to link those tasks to this type of imaging data as well. So as an undergraduate RA, you could potentially be working on an independent project in the lab where you could be involved from kind of the beginning of a research idea all the way to the analysis phase. So typically how it goes is we would talk about different project ideas together. You might do a literature search on that area, summarize what you find, come up with an experimental idea together, and then design that experiment, develop a protocol to run it in human subjects, collect data from participants, which is typically done in front of a computer screen, as I mentioned, with these different tasks, and then be involved in the analysis phase as well. So typically these are obviously going to be happening over a year at least, if not more if you're in the lab for longer. And so that's one option of joining the lab to work on an independent project, but you could also be potentially working alongside graduate students on an existing project. And most of your day-to-day time for the type of lab that we run is involving running the participants on those different tasks that I talked about and doing those neuropsychological tests. Working with young and older adults, since those are the two populations that we study mostly in the lab. You could potentially gain experience with neuropsychological testing. And then we also have weekly lab meetings where we talk about project updates and have guest speakers come that talk about different memory components and all of the things that are kind of related to the lab topics. In addition to individual meetings that talk about your specific project to kind of make sure you're on track with that. So that's a typical sort of undergraduate research experience in the lab, and the types of things that we study and the methods. Does anyone have any questions?
I have a question. How is your lab functioning under the pandemic conditions?
Good question. So we're actually trying to do all of these, well, at least the behavioral studies over Zoom. So it would just be like a one-on-one Zoom session like we are now. And we have a way of presenting those visual stimuli to people through the Zoom session. And so we can try and look at memory from afar, from people just doing these tasks, at home. It's not ideal, but that's kind of the situation that we're in now. And unfortunately, we can't do imaging, the imaging stuff right now either. So that's kind of on pause until it's safe. So.
I was wondering, what is it like to work with participants who are older and who maybe are in the stages of having dementia or Alzheimer's?
Yeah, so mostly in the lab, we do study cognitively normal older adults. So they don't actually have dementia at that point. Randy could probably speak more to patients that have more impairments. But we typically focus on really early, like before it gets really bad, because it's really hard for people even with mild cognitive impairment, which is right before the phase before Alzheimer's disease, for them to be doing these types of cognitive tests, like these complex memory ones that are you really have to have a pretty intact memory to be able to pay attention to small changes in the stimuli, for example. And so it's really hard to be able to test this in people that have, further along memory impairment. So it's really the kind of goal is to detect these changes really early in normal aging before the clinical symptoms really manifest. Any other questions? Thanks.
You can have some clapping.
Thank you. Thank you, Dr. Leal. Yeah, thank you for all the questions. And yeah, we definitely really did enjoy the presentation, so thank you for that. And we can have Dr. Martin next presenting.
Okay. Everybody sees this okay?
Yes,
Okay,
indeed.
good. I've had some weird, I'm using dual screens and it's taken me a while to get this to work right. So it looks like it's doing better than it was. So I'm in the BRC2 in the neuroplasticity lab, and on the 7th floor, I'm the director of that, and Simon Fischerbaum is the assistant director, and now Stephanie's in there, and Brian Denny is as well in that lab. I already told you some of this background, so this is just way long ago. I was a PhD postdoc at Johns Hopkins. I've been here since '82, an Elma Schneider professor. I've been editor of a couple journals, Cognition, Journal of Experimental Psychology, Learning, Memory, and Cognition. And currently I'm the chair-elect of the Linguistics and Language Science section at the AAAS, American Association for the Advancement of Science. And I will also mention that I'm a co-founder of the Women in Cognitive Science Society, which is having its 20th anniversary this year, actually. My research has focused on language and the role of cognitive factors like short-term memory and inhibitory processes in language comprehension and production. And so, as I said in the brief introduction, we've had various methods: behavioral studies of healthy young adults, neuropsychological studies of brain damaged patients, and then neuroimaging of both patients and healthy young adults as well. So I'm going to just focus on 3 projects that are ongoing in the lab. The 2 first ones, we're actively doing research on these. Part of the way we can do that is that we had collected a lot of these data over the years. So we have a fairly big database of patient studies, for example, that we can use for these studies. The third one, if I get to that before running out of time, the third one, we also need to get imaging data. So that is kind of on hold till we can start that up. So the first one is language and working memory recovery from acute stroke to 1 year post-stroke, and looking at behavioral and neuroanatomical changes. The second one is looking at white matter tracks and their relation to different types of working memory. And then the third one is involved in obtaining measures of whole brain connectivity. And this we've done with just healthy young adults, whole brain connectivity and the relation to language and cognitive processes. Okay, so the first one, language recovery from acute to 1 year post-stroke. So we are testing these patients acutely, which means within, well, most of them within 1 week of stroke. Okay, so they have to be left hemisphere stroke, and a lot of them are still in the hospital, and so we have a lab manager that's going in and doing the testing in the hospital. Then they come back at 1 month, at 6 months, and a year. And at each time point, we get a set of behavioral tasks and also neuroimaging data. Main focus of this study is on narrative language production. So this is telling a story. So a lot of studies in psycholinguistics, psychology of language focus on single sentence production, but in this we really wanted to have the patients have to produce an entire story. So we could look at, you know, how many words did they produce, how many words per minute, as well as just individual sentence measures. And then we relate aspects of their narratives to working memory capacities. So what aspects? Well, in the narrative, we look at what's called sentence elaboration, which is just the extent to which they add content words to the sentence. And so within the noun phrase and verb phrase, we're just counting up content words. Mean length of all their utterances, words per minute, and then proportion of grammatical words. So the last one is kind of getting at the extent to which they can produce grammatical structure in the sentences. And then working memory, a main claim coming out of my lab over the years is that in the verbal domain, we have two different kinds of working memory, at least, or probably more. But there's a separate one for phonological working memory and for semantic. Okay, so phonological, we mean speech sounds, memory for speech sounds, and the semantic is the memory for the meanings. Okay, so we're looking at those measures and see how they relate to the narrative. We're also getting single word processing, so just a comprehension of single words, how well can they do that? Because we don't want to have these working memory tasks be impaired just because they can't understand single words. Okay, so we're controlling for that. All right, I'll ask you a question. What's a standard working memory task? Anybody know?
I knew just 1 minute ago.
But now you forgot. Well, a standard one in the literature is digit span, where you just hear a list of digits and then you repeat them back in order. So that's one. That taps mainly into phonological working memory, holding on to phonological information, because there isn't a lot of meaning associated with a random set of digits. And so we use tasks like that to tap into phonological, and then different tasks for semantic working memory. So the questions we're asking are, do these working memory measures predict narrative measures? And so you'd expect this to be the case if language production draws on working memory. Okay, so if you have to hold in mind what it is you're going to say as you plan it, and doing that planning draws on working memory, then to the extent to which the patient's working memory is impaired, they should be impaired in various aspects of these narratives. So we're looking at that. And then we're also looking at, because we're doing this across time, we're looking at, do changes in working memory predict changes in narrative? Okay, so we specifically, for one example, we think that semantic working memory relates to this sentence elaboration. So the greater your semantic working memory, the better you can elaborate noun phrases, verb phrases, put more content into them. And so first we're just looking, do those things correlate? But then we can also look, if you look acutely to 1 month and patients improve on that working memory measure, do they also improve on that specific aspect of narrative production? So that's kind of a stronger test of there being a relationship between these things. The narrative production in phasic patients, they look at the Cinderella storybook. So here's an example of a couple pages from that. We showed that to them. We closed the book and then asked them to tell the story. So we transcribe and score their production. Okay, so we have to be able to transcribe what they've said and then have some methodology for scoring what they came out with, and to get all those measures I was talking about, and then relate their performance to working memory controlling for their single word processing.
The godmother and Cinderella, and it's magic. And Cinderella wants to go to the ball. And the dress and white and the mouse and the pumpkin change up into the carriage and the horses. And
okay, so that's just an example of one of these acute patients telling the story. Just a segment of it. I think this patient did go on, but this gives you a sense of what the patients might sound like, and you know, the various things that I talked about we would score out of this. So this is ongoing, and so student involvement in this could be to learn to score the narrative production, and then once you've learned it, to carry out the scoring and enter the values into databases that we'd be using. Now, learning to score this is kind of an arduous thing. It usually takes people like 1 semester to really learn how to do this, because you never know what the patients are going to come up with, and the instructions seem like they don't always quite apply to what the patients are doing. So, and it's just a lot of sort of technical things to learn about how to do this. But once somebody has done it, then it's nice to get them to stay on in the lab once they've learned it, so that they can continue to do this with additional patients that we are testing. Another part of this project is just creating databases with the narrative and other cognitive measures. The working memory measures, the single word processing, there's also some other measures I haven't talked about, but just putting those all together. And I mean, it takes some time to put this all together. We now have 95 people at the acute stage of stroke, so it's a really large sample for this kind of study. And then we have smaller numbers that have come back at the different time points. And so we mostly so far just scored the data from the acute stage and done analyses on that. And so there's still work going on for the subsequent follow-up stages. And then students have gotten involved in carrying out preliminary analyses of the correlations of the working memory measures and the narrative measures, and then the changes across time points. So one student who did a honors thesis in cognitive science got involved in this at looking at the change from acute to 1 month and relating the change in working memory to some of these narrative measures over that time point, just on the subjects we had up until that point, which I think were about 22. So that kind of thing is something that people can get involved with. Okay, the other one is more looking at the brain structures per se. So looking at the integrity of white matter tracts and their relationship to working memory. So most studies that have looked at the relation of brain damage to working memory have focused on gray matter. Okay, so like looking at the superior temporal gyrus or the inferior parietal lobule and supramarginal gyrus, like how much damage is there to those regions, and does damage to those regions predict performance on working memory? But it's also important to look at the integrity of white matter tracts that connect regions. Okay, so the gray matter could possibly be preserved, but the connections between regions might be damaged. Does that make sense to everybody? Yeah, so, you know, the speech perception region, let's say superior temporal gyrus, and the region for maintaining phonological formation, which is partly the supramarginal gyrus, those might be preserved, but if the fiber tracts that connect them are lost, then patients would be able to take what they've heard and perceived and store it in working memory. Okay, so it's important to look at these white matter tracts as well. So this project is being carried out with chronic stroke patients. So we have collected data, we collected scans, and we've collected behavioral data, a lot of behavioral data on chronic stroke patients over the last 15, 20 years or so. And so we have about 60 of them now in this database where we have their scans, and we also have DTI imaging that allows us to do this fiber tract tracing, and tons of behavioral measures, including working memory measures. So our screening battery for these chronic patients takes about 4 days to administer 2 hours a day. So everybody, we've got like 8 hours of testing at least on them. And so there's a whole lot of other issues that could be looked at once we start tracing these tracts. So have people heard of this diffusion tensor imaging? Raise your hand if you haven't heard of it. No? Okay. So what you're seeing here is just sort of a map of a lot of different tracts. So the white matter are the axons coming out of the neurons, okay, and they come together and form these tracts that connect different gray matter regions, so activation can be sent from one region to another and back. So you've all seen the scans that might just show you know, the neuroanatomy or show activations. Okay, so these are not activations. These are, it's really a structural imaging technique that the sequences that are used in the scanner, they're looking for the flow of water molecules. And if they find regions where all these molecules are going together, that probably, and they do that along these white matter tracts. They'll do that along the site of the white matter tracts. That will give you an indication of where a white matter tract is by that kind of imaging. So I won't say more about that, but so these are showing some of the tracts that people have already said are probably related to some aspects of language. So you're seeing one here, inferior longitudinal fasciculus, here inferior frontal occipital, here arcuate, and here the uncinate. And so those are some we would be looking for. And this just shows you brain damage. So this is for a patient with aphasia. So aphasia is a disruption of language after brain damage. So like the patient you heard in that example of the Cinderella is an aphasic patient. And you can see like this green fiber tract, this inferior frontal occipital, fasciculus. It's right here, and the patient is much skimpier. There's hardly any fibers compared to in age-matched controls. Okay, so, and then this also, this is the arcuate fasciculus, and you can look at that for the age-matched controls. And so what people are learning is how to track these fibers. So there's methodology for doing that. And then get various measures out of that about how preserved this is. So how many strands are there? There's something called FA value, which is just how straight those water molecules are proceeding. So, once we have done this kind of tracing for the patients, then we could look, well, is the integrity of the arcuate, the number of fibers, let's say in the arcuate, does that relate to working memory, and which kind of working memory? Okay, so that's what we hope to do. We're just doing this tracing right now. So the student involvement, I don't know, we got like 7 students right now that are doing this. So, I mean, this is something that can be done now because we have all this data that's piled up over the years. And so they learn to segment these tracks from DTI images. It was a workshop, and then they're getting supervision from graduate students. And getting these tract integrity measures from patients and controls entered into database, and then potentially could do preliminary analyses of correlations of tract measures with working memory measures. Okay, so I think I've talked enough. I seem like I'm already it's almost 5:20. So I'll just zip through project 3 and not talk about it. We can't really do this right now anyway, because we aren't doing scanning yet. So it's getting resting state activity and calculating brain modularity and relating that to cognition, but I won't talk about that. So, and the behavioral measure we're doing is getting eye tracking. So tracking people's eyes while they are reading. And so anybody that got involved in that would learn the eye tracking, as well as getting involved in the resting state analyses. Okay, so that's it. I will stop there. And have my email address up there if you need to get a hold of me. So do people have questions for me?
Yes, Dr. Martin, I was hoping to ask one quick question. As you mentioned, the integrity of the white matter tracts are really important to language and working memory performance, and you typically study stroke patients. I was wondering if there were other ways in which the integrity of the white matter tracts may be compromised. For instance, could it be something that's inherent or innate at birth, or does it typically come from a stroke?
Well, I mean, even healthy individuals will differ in those measures of the white matter tract. And so, you know, in our work so far, just looking at the controls, you can see a really wide range of thickness, you know, FA values within the controls. So there's definitely individual differences in healthy individuals. These controls were older adults, but I'm assuming you would find the same thing in younger adults as well. And so like everywhere you look, there's individual differences. They're just going to be less dramatic than you might see with the patients, where some tracks just aren't going to be there at all. Whereas maybe for the controls, they mostly will be there, but even for that, we found that we were looking at that arcuate fasciculus in the right hemisphere for controls, and some of them just didn't have it. So that's like, I was really surprised when we first saw that, but apparently that's not unique to the controls we tested, that sometimes it's just not there and it shows up more strongly in the left. So I don't know if you guys have heard this yet or not, but back when Wernicke was talking about language in the brain, he claimed that Wernicke's area and Broca's area were connected by the arcuate fasciculus. So that was, in his model, a major component of language processing. So yeah, maybe it's just more solid and consistent on the left, but still shows up in some people on the right.
That's great.
Yeah, you could, I mean, people have done and can do studies with just variation amongst healthy people.
That's great. I didn't realize there was so much variation within the control group even, so that's great to hear. Thank you.
Yeah, it was a surprise to me too. Anything else?
I was just curious, for the first project, when the patients are giving the narratives, do you have to use a different story each time? Or is there any chance that their memory from, say, their first production at the acute time would they remember the story to be able to produce it better the next time? Or does that not really matter?
Yeah, I don't think it makes that much difference because it's such a long time between the first time they say it. It's at least a month until their first visit back. So I don't think it would help. We just use the same one each time because it's kind of a standard thing people have used in the literature, and this scoring method that's been developed has been used on that story specifically. And so just to relate to other findings in the literature, it's nice to be able to do it the same way. You wouldn't have to, but if we use something else, we'd want to make sure it was equally evocative of a story. We wouldn't want to change and then whatever we use the next time, it doesn't really elicit that much out of them. So that's another reason just to stick with the same thing. Now the patients they're generally getting better over 1 month and certainly up to 6 months. So there's a lot of spontaneous recovery in that time period. So they're improving and they're getting better, but I don't think it's because they remember what they said before. I don't think they remember what they said before. We asked them a couple hours later. They, had a lot of them had trouble getting it out, and it was it's a struggle. The one you heard wasn't so bad, but some are worse. Where they're just producing single words rather than sentences. Anything else? No? Okay, well, we don't really need anybody else this semester. We've got so many people working in the lab. I think because we had all these data piled up, and so, we had a lot of stuff people could work on that didn't require getting new data, but we may need new people by the next semester. People may not want to continue or, move on to some other project, even within our lab. So if you're interested, you could check back like mid-November or something to see if anything's open for next semester.
Thank you, Dr. Martin. And finally, we have Dr. Pitkow. We're a little close to 5:30, but that's okay. We can go over a little bit, so no worries.
Yeah, I'll keep it super short. I have more than enough to cover, so I'll just show some pretty pictures. Let me share my screen. All right, let's see, hopefully this will work. Nope, wrong screen. Hang on. Let me share the other screen. Okay, how's that? All right, and I'll put you on gallery view. If you would kindly turn on your cameras so that I can see your alert faces and know when it's making sense and when it's not, that would make my life much better. It's no obligation, but so thank you. I appreciate that. Yeah, I'm interested in principles of neural computation. And so I like to think about what are the core elements of intelligence, which I think of as kind of the union of statistics and flexibility and compositionality. And if you put all those together, you get something that is able to understand the world to some degree. And so this is a major question present right now as computers are becoming smarter. So we use a lot of information from computer science. Here's a picture I drew that kind of evokes the connection between neuroscience and machine intelligence. So we have a smart brain and we have a smart machine, but the machine is nowhere near as smart as we are. And so trying to figure out how we can take principles from neuroscience and get smarter machines, and conversely, and test those ideas in machines. Conversely, take ideas from machine learning and see if we can use them as theories and predictions for neuroscience. And so then there's this strong interplay between data and theory that forms the kind of core way of mediating between these two ideas. So my lab does a lot of math. This is an actual whiteboard in our lab that I just inverted. And we collaborate with people who work on all different scales with worms and mice and monkeys and humans. We have some core foundational hypotheses that the brain is well adapted to the world and that it has some model of the world that's matched to it. And we'd like to figure out what the model is, what assumptions are there, and how does the brain actually draw inferences and choose good actions. This gives us predictions about neuroscience, which are kind of fun because they can, if you have a mental model of the world, you can augment your ability to understand what the neurons are related to. So in sensory neuroscience, you're looking at the brain activity, you measure stuff, and then you measure the visual world or the auditory input or some words or something outside in the brain that you can measure. And you see what's the relationship between those two. Or you might do motor neuroscience and you might look at how the muscles change with respect to brain activity, or maybe you're producing speech, different kinds of outputs. But if you want to understand thoughts, you might be able to measure brain activity, but what are you going to compare the brain activity to? Right, so that becomes a challenge. So that's where we make models. We develop theories of what you must be thinking that could explain how you transform your sensory input into motor output, which is what we're able to see, and then connect that to the brain. This means that we use a lot of control theory, which allows you to think about the loop between the world, giving rise to observations, and then influencing the brain, which has some beliefs about the world, and then go back and create actions which interact again with the world. So there's this loop here, and we try to understand, look inside the box. And we do that by picking different tasks. We have some, like an example set of tasks is we have monkeys playing games in virtual reality. And like this one's a firefly catching task. So you're supposed to steer with a joystick to catch to land on that circle there. This is what it looks like if you had a mountain background and if the target stayed on, but we make it harder. And so you have to navigate to this unseen target. And monkeys can actually do this. They do this pretty well. Or people do it too. We do that with people. And I don't do any of these experiments. None of the experiments that I'll talk about here are things I've done. I'm just a theorist. But I collaborate with these experimentalists who have some amazing experiments. Like, for example, recording wirelessly from monkey brains as the monkeys move around and play different games in an environment where they can climb on the walls and swing from the ceiling and get food from these little dispensers. And yeah, okay, so that's one kind of category of projects that we have. Another category which harkens back to the thing I was talking about in the beginning is trying to understand computational functions of the brain in terms of machine learning ideas. And how can you get really good, smart machines that are inspired by or derived from the brain? And so we combine constraints from different aspects of what brains do. It can be doing tasks like demanding that an algorithm perform the same task that a human or an animal can do, or use the neural activity. Like these ellipses, imagine these are constraints, and the intersection of the constraints gives you a good algorithm. So you can have strong generalization when you combine good signals of neural data, good descriptions of biological architectures, and lots of different tasks that they have to do. Those constraints may give you something smarter than any subset of those things. So we try to put all those together. And we do this with some amazing data. We have this historic data set that we get to work with that's collected by my colleagues at Baylor. Andreas Tolias's lab has collected this data, which is neurons from a cubic millimeter of a mouse brain while it's watching a video. And then we slice the brain into 25,000, this cubic millimeter, I should say. We slice the cubic millimeter into 25,000 sections. And then using those sections, you can do a 3-dimensional color by number to get the exact morphologies of these neurons at the nanometer scale. You can see individual synapses and vesicles. So this is really an impressive thing. And here, if you overlay with them the actual functional responses that we recorded to the movie, then you can try to relate the structure to the function. This is some of that data. 75,000 neurons in a single animal recorded with calcium imaging. So what does this mean? That's what we're after. We're trying to figure out what this means. And so we have some models that, that we go through and try to relate these two, and I'll kind of skip over this in the interest of time. And go to, oops, what happened to my, this autoplay? I'm jumping to, let's say, to here. Okay, so we're trying to like interpret a video as the animal sees this video. We imagine that the neurons or populations of neurons are passing information back and forth that allow it to get better interpretations of this kind of sensory input. And so what are good algorithms of that? And the math that we use for this comes from a field called probabilistic graphical models. It's a way of describing knowledge. Okay, stop. I want to go to the next slide. There we go. Probabilistic graphical models allow you to describe the structure of the world in a way that doesn't allow absolutely anything to happen. It gives you this structure, and you can assume structure and you can learn structure. So this is kind of a fundamental equation that we use. And of course, all the details are in what do these symbols mean. And then once you have a representation of the world, if you want to figure out what to do, you have to recognize what's important right now and ignore or account for everything else. And so that's a different operation called marginalizing. So we use these types of mathematical models to try to understand what might the brain be doing and use that as a hypothesis that we could test. So these are hard things to do, but that's where we have fun doing theory. We also make games, games to try to teach mathematical concepts or other neuroscience concepts. I have a student who works with me who's developing computer games that you can interact with, and we develop them as open source. So that's one thing that you might be interested in working on. So there's some research projects that we do, and then there's some sort of teaching projects that we do that, of course, when you're doing research and doing, you often have to learn concepts that are new, and you learn by doing them, but you can also learn by teaching them. And so developing these kind of games allows, I mean, certainly it's taught me a lot about how to understand neuroscience concepts at a deeper level. So I collaborate with a lot of people. As a theorist, I need to convince people to do experiments and get data from them and have a rich interaction. And so these are some of the PIs, the principal investigators, the faculty and the professors that I work with. And these are people who work with me in the lab, which is the laboratory for the algorithmic brain. And so we have a couple of undergrads, Noshin and Adit, who have been doing some awesome research. I'm happy to talk about that, their stuff if you want. We have a few graduate students and postdocs, and you know, maybe your name goes here too. So that's my brief synopsis of what we do in the lab. And I am happy to take questions and draw pictures, and we're already time over, so I guess we'll keep it short. And my email, which I'll put in the chat if you're interested, you can contact me, is xaq@rice.edu. So there it is.
So yeah, I had a question about, so I know you mentioned how we're able to use these brain models to sort of translate into machine learning and like how to, you know, create more efficient machines. So I'm wondering like how, I guess just on a surface level, like how do you use that kind of information and translate that to like modern machines and things like that?
So you could say that the proof is in the pudding. If you have a theory about the way the brain works, does it actually work that way? Try it. Like, you think that vision works by this algorithm. Okay, try it. Here's a benchmark task. See if you can recognize cats and dogs using your model. So there's a lot of stuff that's been done in that direction now. You probably have tons of it in your phone, or you certainly do if you have a phone. And so we try to compare what we do to what those algorithms do. So literally we do the same machine learning tasks, but with algorithms that are inspired by brains or directly derived from brains. One example is you have a representation that emerges when you try to train a neural network to do a thing. What if you also encourage that neural network to make activity patterns that look like the brain that you just measured? Does that help? Does that hurt? Turns out that in our hands, it helps, and it makes it more robust, which means that it generalizes better. It's able to take problems that seem like it's outside of the training set. And those are things that often fool conventional algorithms, but these brain-derived ones, they do better. So that's the way that we try to connect those two. We really test it in the same tasks that machine learning people test theirs on.
That's great, thank you.
Who here likes math? Yay, math. Number one most important math to know for, I think, brain science in general, and maybe many other things, is linear algebra. So if you've not taken linear algebra, I encourage you to take it. That's basically like adding, subtracting, multiplying, and dividing when you have more than one number at a time. And when you have a brain, you have more than one number at a time. Who wants to see monkeys playing virtual reality video games? Come on, that's fun, right? Although these are fraught problems, right? Animal testing is something that bothers me a great deal. I am absolutely complicit. Okay, well, on that optimistic note, if nobody has any other comments or questions, you have my email. You can contact me. And Amerta, thank you for the invitation. There was one other person who invited me. I forget who that is, wasn't it? Or was it? I just would like to say thanks to them too. Maybe it was just you. I remember two names, but I may misremember. Well, thank you everybody for spending the time with us.
Yeah, thanks everybody.
Yeah, I want to thank all our panelists and also to everyone who was able to join today. This is really great information for a lot of our undergrads who are interested in just learning about research and what it looks like. So thank you all for presenting your wonderful research to us. Thank you.
Take care.
You
Bye.
too.
Thank you for inviting us.
Of course.
Bye.
Bye, everybody.